Okay, so we'll quickly start the molecular biology. You can quickly give your feedback if you can hear me, you can see me, and if the iPad is clearly visible. Okay, so we are starting the molecular biology session. We'll ignore what happened yesterday. We'll assume that yesterday's session did not happen, and we start afresh from our beginning of molecular biology. So, like I told you, we'll be talking about two things. First, we'll talk about the properties. First part, properties. And when I say properties, here we'll be talking about first the nucleotides, and then we'll talk about the nucleic acids. Obviously, when I say nucleic acids, it means the DNA and the RNA. Once we discuss the properties, then we will discuss the metabolism. And when I say metabolism, obviously it means biosynthesis as well as the breakdown. And in this metabolism, we'll talk about first the purines and pyrimidines, the nitrogenous bases. After that, we'll talk about the DNA synthesis, we'll talk about the RNA synthesis, and we'll talk about the protein synthesis. Time permitting, we'll try to cover the regulatory model called the lag operon, and wherever applicable we'll also be simultaneously talking about the techniques. So these are the things that we're going to cover in our molecular biology section. All right, so let's start with the topic of nucleotides. So what are nucleotides? When I say nucleotides, what do I mean? Please note, the nucleotides themselves are made up of the three subunits. At the center, we have the pentose sugar. At the center, we have the pentose sugar, to which the nitrogenous base is attached, and on the other end, the phosphate is attached. So pentose sugar attached to the nitrogenous base and phosphate will make up the nucleotides. Sometimes we find only the pentose sugar and the nitrogen base when we call the molecule as the nucleoside. So remember, when I say nucleoside, it means only the pentose sugar and nitrogenous base. But when I say nucleotide, it means the nucleoside attached to one or more phosphate. So this is a very important distinction to keep in mind. It is very frequently asked in your exam. You should remember, nucleoside means only the sugar and the nitrogenous base, whereas when I say nucleotide, it is the sugar, nitrogenous base, and the phosphate. Okay, so what we'll do, we'll look at the subunits, the nitrogenous base and the pentose sugar separately. Once we have done that, then we come back to the combined properties of the nucleotides. Okay, so starting off with the nitrogenous bases. So now we are going to be talking about the nitrogenous bases. Now when I say nitrogenous bases, immediately a few points should come to your mind. For example, the nitrogenous bases are heterocyclic compounds. They are heterocyclic compounds. They have what is known as, okay, see, first of all, they are ring structures. That is why the term cyclic is there. Typically in the ring structure, in the ring we only have carbon, but here in the ring there will be atoms other than carbon, and therefore the term hetero is used. And if you look, you'll find that the ring contains nitrogen. The ring contains nitrogen. That's why we call it the heterocyclic compound. We call it the heterocyclic compound, nitrogen containing ring structures. Whenever we have the ring structures of carbon, we've already seen in case of carbohydrates, they have a planar character, meaning some of the additional groups will be above the plane of the ring and some of the additional groups will be below the plane of the ring. So they have what is known as the planar character. They have what is known as the planar character. And lastly, we have tautomerism. Tautomerism means there is interchange of functional groups. There is interchange of functional groups due to migration of double bonds. I'll show you an example. See what happens. There is alternating double bond in the structure. There is an alternating double bond in the structure. And to this double bond, there can be groups which are attached. For example, the hydroxyl group. Groups like hydroxyl group may be there. Now, the oxygen which is there attached to the ring, it has a tendency to pull the shared electron towards itself. So what happens? This shared electron is pulled towards oxygen so that the structure becomes like this. So what do we note? The interchange of functional group. The hydroxyl group has now become the keto group. The hydroxyl group has now become the keto group. There is a migration of the double bonds. There is migration of the double bonds. The other double bonds in the ring will also shift when the oxygen pulls the electron. Therefore, you see the adjoining bonds become double bonds. And this change in the functional group will obviously change the property of the molecule, because the functional groups are important for the chemical properties and to some extent a bit of physical properties also. Therefore, there is this interchange of functional group. Typically, the hydroxyl groups gets converted to the keto group, and the amino groups get converted to the imino groups. So this is the two common type of tautomerism that we get to see in the nitrogenous bases, the hydroxyl to keto and amino to the imino group. Now, the nitrogenous bases are broadly grouped together into two categories known as the purines and the pyrimidines. Two broad categories, the purines and the pyrimidines. Purines and pyrimidines, I told you the common purines and pyrimidines are the adenine and guanine. The common pyrimidines are the cytosine, uracil, and the thymine. Adenine and guanine are found in both DNA and RNA without fail, whereas the pyrimidines, among the pyrimidines, the cytosine is found in both DNA and RNA. The uracil is found in the RNA, whereas thymine is found in the DNA. One of you had asked about thiamine being vitamin B1, so please note the spelling of thiamine, that is vitamin B1, and the thymine, that is the nitrogenous base. I know that there is some confusion, but you'll have to make note of this. Thymine is found only in the DNA, with a certain exception of transfer RNA. However, there the thymine is a modified nucleotide. We'll talk about this when we are talking about the tRNA. In that case, the thymine is a modified nucleotide. The thymine is a modified nucleotide there. Okay, so this is about the purine and the pyrimidine. The second thing that you have to know is the purines are actually quite big molecules. They have two rings attached to each other. One of them is a six-membered ring. One of them is a six-membered ring, and the other one is a five-membered ring with alternating double bond. Alternating double bond. With alternating double bond being there in the purine, whereas pyrimidine is having only a single ring, which is a six-membered ring. It has a single ring, which is a six-membered ring, again with alternating double bond. With alternating double bond. If you look at the numbering for the purines starts from here, where we have the N1, C2, N3, C4, C5, C6, N7, C8, and N9. This is slightly different than what we see in the pyrimidines. In the pyrimidines, the numbering starts from here and goes in the opposite direction. N1, C2, N3, C4, C5, and C6. So what do you find? If you look at the purine, the six-membered ring is numbered anti-clockwise, whereas in the pyrimidine, the numbering is clockwise. This is a very important point to keep in mind. The purine and pyrimidine are numbered differently. Their numbering pattern is not the same. This statement is given in the question: which of the following is correct, which of the following is not correct. So you have to remember the numbering pattern of purine and pyrimidine is not the same. They have a difference in the numbering pattern. The purines have two rings attached to each other, having a total of 9 atoms in the ring, out of which 4 are nitrogen, 2 in each ring. The pyrimids are made up of 6 atoms, out of which 2 are nitrogen. Because it has a single ring, so it will have only 2 nitrogen, because there are 2 nitrogen per ring. So remember, the purine, the smaller name is actually a bigger molecule. The pyrimidine larger name is actually the smaller molecule. The purines have 2 rings, the pyrimidine will have the single ring. The common purines are the adenine and guanine. The common pyrimidines are the cytosine, uracil, and the thymidine. So these are the nitrogenous bases, which are heterocyclic compounds. They have a planar character and they show the phenomena of tautomerism. Please let me know if the feed is better now. If you are able to see it correctly now, just let us know. Would like to hear from you whether it is coming on correctly now or not. Okay, is it better now? We are hoping that is better now. Coming to the pentose sugar, coming to the pentose sugar. The pentose sugar can either be a ribose, or it can be the deoxyribose. And as you already know, the ribose will be found in the RNA, and the deoxyribose will be found in the DNA. The deoxyribose will be found in the DNA. Alright, so we have the RNA and the DNA having the different type of pentose sugar, the ribose in the RNA and the deoxyribose in the DNA. And the name itself should give us a hint, because the RNA says ribonucleic acid and the DNA says the deoxyribonucleic acid. Alright, so the difference in the sugar is there. If you look at the structure of the sugar, in this case it is a ring structure. It is a 5-membered ring with the 5th carbon being outside the ring. The 5th carbon being outside the ring. Carbon 1, carbon 2, carbon 3, and carbon 4 make up the ring along with oxygen. Which type of ring is this? Is it a furanose ring or is it a pyranose ring? Which type of ring is this? Is it a puranose or the pyranose ring? pyranose or the furanose, I'm sorry, pyranose ring or the furanose ring, which type of ring are we looking at? Remember, this is the furanose ring, the 5-membered ring or the furanose ring. And to this structure at position 1 of carbon, the nitrogenous base is attached. Recall that the nitrogenous base we had just seen is a ring structure. The sugar in this case is also a ring structure. How do you distinguish between the two? If I say, let's say, I say carbon 2 has a hydroxyl group. So whether I'm talking about the sugar or I'm talking about the nitrogenous base, both of them are going to have the carbon 2. Both of them are going to have the carbon 2. So please note that this dash, also sometimes known as prime, is used to distinguish the atoms in the sugar from the atoms in the nitrogenous base. This is how we distinguish the two. When I say carbon 2 dash, I'm talking about the sugar. When I say carbon 2, I'm talking about the nitrogenous base. For example, we say the orientation of the DNA, orientation of RNA is from 5 dash to 3 dash end. So in this case, we are talking about the linkages between the sugar, not between the nitrogenous bases. The orientation is defined by linkages between the sugars. We'll see later. So that is how we come to that. Okay, so to distinguish between the sugar and the nitrogen base, we use the annotation prime. And please note that the deoxygenation is seen at carbon 2 in the deoxyribose. So the correct name should be 2 dash deoxyribose. For the DNA, the name of the sugar would be 2 dash deoxyribose. The nitrous base being attached at carbon 1 dash. The nitrous base is always attached at carbon 1 dash, but for purine and pyrimidine, the point of attachment is different. The purines are attached via N9. The pyrimidines are attached via N1. The point of attachment for nitrogenous base with the sugar is different. At the sugar, they are always attached at carbon 1 dash, but in case of purine, that linkage is via N9. In case of pyrimidine, the linkage is via N1. So when you attach the sugar to the nitrogenous base, you get the nucleoside. We get the nucleoside. In case of nucleoside, from the nitrogenous base, the adenine becomes adenosine. The guanine becomes guanosine. Alright, so like that we will have the changes for the nitrogen bases and they are getting converted to the nucleoside. Okay, so this is how the nucleoside is formed. When we come to the nucleotide, recall we had the phosphate in the nucleotide. The phosphate is commonly attached to 5 dash. However, it may also be attached to the 3 dash end. So there are two potential points of attachment. Either it can be attached to the 5 dash end or it can be attached to the 3 dash end. More commonly, it is attached to the 5 dash end. The phosphate is more commonly attached to the 5 dash end. Alright. In addition, the number of phosphates is also variable. It may be 1, it may be 2, or it may even be 3. You know that we have the adenosine monophosphate, the adenosine diphosphate, and adenosine triphosphate. All of them are nucleotides. So the number of phosphates is variable. It may be 1, it may be 2, or it may be 3. All right. However, the second and third bonds of the phosphate will be different from the first bond. Why? The first phosphate is attached to the carbon, but as you can see, the second and the third phosphate are attached to another phosphate. So obviously the nature of bond will be different, and very frequently you are asked about the different types of bonds in the nucleotide. So we'll quickly label the different bonds that we see. The bond between the nitrogen base and carbon dash is the beta N glycosidic bond. It is the beta N glycosidic bond. The bond between the sugar and the first phosphate, it is an ester bond, whereas the bond between the different phosphates, the bond between the different phosphates is the acid and hydride bond. These acid and hydride bonds are under a lot of pressure because the phosphates have negative charge. The negatively charged molecules will repel each other. This bond is under great tension, and when you break this bond, all the energy which is trapped in this bond will be immediately released. That is why cleavage of phosphate provides us with free energy which we can use to drive the other reactions which normally will require extra energy. They can be compared to what is commonly known as the high energy phosphate. Okay, so acid and hydride bonds easily broken to give us energy to drive the other reactions. Till now we talked about three bonds: the beta N glycosidic bond, the ester bond, and the acid and hydride bond. Okay, this is about one nucleotide. Now when you talk about the nucleic acids, obviously the multiple nucleotides will be attached to one another. So what I'll do, I'll draw another nucleotide below and we'll try to link them together. Another nucleotide, and we'll try to link them together. Okay, so this will be C1 dash, C2 dash, C3 dash, C4 dash. To the C1 dash there is attachment of the nitrogenous base. Now when the two nucleotides join together, the second incoming nucleotide will come and get attached at the carbon 3 dash position. It will come and get attached to the carbon 3 dash position via the triphosphate which is there at the C5 dash. During the linkage formation, the pyrophosphate part will go out. The pyrophosphate drives the pairing. The energy is provided by the pyrophosphate, which further cleaves into two inorganic phosphate, and thus the bond is formed. You can see the phosphate acts as a bridge between the two sugars linked from 3 dash to 5 dash. Two things to note. Number one, look at the bond. The bond between C5 and phosphate is an ester bond. We already said. Exactly same bond is there between the phosphate and C3 dash of the first sugar. Therefore, what we are getting is actually a diester bond. And because the phosphate is the bridge, we call it the phosphodiester bond. So the bond between the two sugars of the different nucleotides is known as the phosphodiester bond. The bond is the phosphodiester bond. Second thing you note that the 3 dash end of the second nucleotide is free. This is where the next third nucleotide will come and get attached. So always the attachment of the new nucleotide, whether it is the DNA or the RNA, will be at the 3 dash end. So one 3 dash end will always be free. And if you look at the orientation of the nucleic acid, at one end you will have the 5 dash end which is free. The 5 dash end is free, and on the other end where the new nucleotide is going to come and attach is the 3 dash end. So when we talk about the orientation of nucleic acid, whether it is the DNA or the RNA, we say the nucleic acid is oriented from 5 dash to 3 dash. for all nucleic acid, whether it is the DNA or the RNA, we will read the sequence from 5 dash to 3 dash. We'll assume that the 5 dash end is the first nucleotide, and subsequently the nucleotides have been added in the 3 dash direction. So all nucleic acid, whether it is a DNA or the RNA, have to be read from 5 dash to 3 dash direction. From 5 dash to 3 dash direction is the traditional way in which we have to read the nucleotides which have been attached. Okay, so now we have the nucleic acid chain which is being formed here. Till now we have talked about 4 bonds. The fourth bond was the phosphodiester bond. The fourth bond was the phosphodiester bond. Same manner, and more nucleotides can get attached, and the structure that you are seeing here, the sugar, the phosphate, next sugar, sugar, phosphate, and the next sugar, the structure that you are seeing here, this is called the backbone, the backbone of nucleic acid. And the alternating sugar and phosphate makes up the backbone. Remember, the nitrogenous base is not there in the backbone. If you're looking at a double stranded molecule, whether it is a double stranded DNA or the RNA, we can expect a similar backbone running here, but this backbone will have an opposite orientation. If the first one was running from 5 dash to 3 dash end, the second one will have the 3 dash to 5 dash end orientation. It will have the opposite orientation. This is so to ensure that the nitrogenous bases come to face each other. If the orientation of the second nucleic acid is also 5 dash to 3 dash end, in that case our nitrogenous base would have been oriented on this side, and then the two nitrogen bases will not come close together. So by arranging it in an anti-parallel fashion, by running it in opposite direction from the first nucleic acid, we ensure that the nitrogen bases have now come together, they are facing each other, and in this position they can be linked together by the weak hydrogen bonds, which may be either 2 or 3 in number. Alright, so the two nucleic acids are joined together by the weak hydrogen bond between the nitrogenous bases. They are joined together by the weak hydrogen bonds between the nitrogenous bases. This bond which I have just shown is one of the most frequently asked bonds in the exam. What is the nature of the bond between the nitrogen bases of a double stranded DNA? This is very commonly asked. It has also been asked as an image based question. It has been asked as a straight line question. It has been asked indirectly that two strands of nucleic acid are held together by. So there also you have to mark the hydrogen bond. So you should be very clear that the two strands of nucleic acid are not held together by any covalent bond, rather it is a non-covalent hydrogen bond. Alright, so I'll quickly summarize the different bonds that you get to see in a nucleic acid. We started with the beta and glycosidic bond, which is seen between the sugar and the nitrogenous base. Then we have the ester bond, which is seen between the sugar and the first phosphate, followed by the acid anhydride bonds, which are seen between the second and the third phosphate. Second, third phosphate, when they are released, will give us a large amount of energy. The two sugars are linked together by the phosphodiester bond. The two sugars are linked together by the phosphodiester bond. All these 4 bonds are covalent bonds. The first 4 bonds are covalent bonds. Subsequently, the two nucleic acids are joined together by the non-covalent hydrogen bonds. They are joined together by the non-covalent hydrogen bonds. So the 5 bonds that you have to keep in mind within a nucleic acid. Now we said that the two strands of DNA are held together by hydrogen bond between the nitrogenous bases. But one question which should come to mind is the pairing between the nitrogenous bases random, or does it follow a pattern? Interestingly, it follows a pattern, and this pattern is described by what you very well know as the Chargaff's rule. The Chargaff's rule states that adenine will pair with guanine. Sorry, adenine will pair with thymine. The guanine will pair with cytosine. The guanine will pair with the cytosine. If you talk about the Chargaff's rule in the double-stranded DNA, what follows is like this. See, we have the double-stranded DNA. So every time there is an adenine on one strand, we'll have a thymine on the second strand. If we have adenine on the second strand, we are going to have a thymine on the first strand. So in double-stranded DNA, the amount of adenine will be equal to amount of thymine. Similarly, the amount of guanine will be equal to the amount of the cytosine. Alright, so this is the interpretation of the Chargaff rule for double-stranded DNA. If you add the two sides, what you get is adenine plus guanine is equal to thymine plus cytosine. Now you know adenine and guanine are the purines. Adenine and guanine are the purines. The thymine and cytosine are the pyrimidines. So in double-stranded DNA, the amount of purine is equal to the amount of pyrimidine. The amount of purine is equal to the amount of pyrimidine. So the two fundamental Chargaff rule are the adenine equal to thymine and guanine equal to cytosine. A follow-up is the amount of purine equal to pyrimidine in the double-stranded DNA. But the relationship do not stop here. We can extrapolate these relationships. For example, if adenine is equal to thymine, this means the ratio of adenine to thymine will be 1, or the ratio of thymine to adenine will be 1. Similarly, the ratio of guanine to cytosine will be 1, which is equal to the ratio of cytosine to guanine. The ratio of adenine plus guanine to thymine plus cytosine is also equal to 1, which is equal to the ratio of thymine plus cytosine upon adenine plus guanine, and the ratio of purine to pyrimidine is equal to 1, which is equal to the ratio of pyrimidine to purine. These are some extrapolations of the relationship described by the Chargaff's rule. We can make more and more such permutations and combinations, and it becomes difficult to keep them straight in your head. What is the correct relationship? So in the exam, and this relationship is asked very commonly. You'll be given various combinations and you'll be asked which of them is correct, which of them is false. For example, I'll give you one example. A plus C is equal to T plus G. Is this right or wrong? What do you think? A plus C is equal to T plus G. Is this right or wrong? What do you think? I'll write this a little bit below so that it becomes easier later on to discuss. A plus C is equal to T plus G. You have to tell me whether this is right or wrong. Very quickly, tell me whether this is right or wrong. I'll just try to describe how to approach this type of question, and then we'll go back whether it is right or wrong. I'm not saying whether it is right or wrong, but I'm just saying you may be right. But let's see how to approach this sort of question. Now, I told you it becomes difficult to keep the various relationships in the mind, which one to remember, which one to forget. So what you do, obviously you remember this relationship, A is equal to T. Let's say that it is rule number 1. You remember the rule number 2, G is equal to C. Let's say this is rule number 2. Now, whenever you find an equation using these relationship, whenever there is a talk of Chargaff rule, whenever there is some equation where adenine, thymine, guanine, cytosine are being put in, and you are being asked which of the following is correct, which of the following is false, in that case, what you do, wherever you find adenine, replace by 1. Wherever you find thymine, replace by 1. Wherever you find guanine, replace by 2, and wherever you find cytosine, replace by 2. Alright, if you do this, and still the two sides are equal, that means the equation is right. If you do this but the two sides are not equal, this means the equation is false. Please note, if the two sides are equal after putting the value, the equation is right. If the two sides are not equal after putting the values, then the equation is not correct. Let's test it in the question that I gave you. Adenine is 1, thymine is 1, cytosine is 2, guanine is 2. In other words, the left side is a total of 3, the right side is a total of 3, and this implies that the given equation is correct. If both sides are equal, then our given equation is correct. Now you'll say, sir, but how is it possible? It is possible because, see, we said adenine is equal to thymine and guanine is equal to cytosine. They can be on any side. And if you add the two, A plus C is equal to T plus G. But we don't have to do this jugglery in the exam. What we have to do, we just replace the value by 1 and 2. A and T are 1, G and C are 2. Replace the values, compare the two sides of the equation. The two sides are still equal. After putting in the values, the equation is correct. If the two sides are not equal, then the equation is not correct. For example, a question can be like this: the A plus T is equal to G plus C. True, false. A plus T is equal to G plus C. True, false. So what do you do? We quickly put in the values. A is 1, T is 1, G is 2, C is 2, and the equation is trying to tell us that 2 is equal to 4. No, this is not possible, and we directly say that is false. So this is how you are going to approach the questions on the Chargaff rule. Replace the alphabets by numbers, and then you can quickly check the validity of the equation. You don't need to memorize much. The two equations that we mentioned you already know. You only have to use those two equations and replace the values by 1 and 2. This is the Chargaff's rule. Very frequently the question on Chargaff rule is coming, and you'll be asked about what is correct and what is not correct. So this point should be clear in your mind, how to approach the question on the Chargaff rule, how to approach the question on the Chargaff rule. Okay, so let's go back to the nucleotides. I hope everyone is clear about the Chargaff rule now. I hope everyone is clear about the Chargaff rule now. If you have any doubts, please let me know so that we can quickly carry it forward and move on. If there are any doubts, quickly let me know. Okay. So now that we have talked a little bit about the structure of the nucleotides, let's quickly talk about the properties of the nucleotides. Properties of the nucleotides. The first and foremost, the nucleotides are building blocks. First and foremost, they are building blocks of nucleic acids. You already know this is the most important property of the nucleotides. They are the building block of nucleic acids, but besides that they also act as energy transducers. They act as energy transducers. For example, ATP, GTP. What I mean by energy transducer, in the reaction, if a lot of energy is being released, they will be captured by the nucleotides. We had seen the substrate level phosphorylation in the carbohydrates. All right, so that is trapping of energy, and where energy is required, that ATP will break down and release the energy. For example, in the urea cycle, we had seen the ATP is providing the energy. In the glycolysis, in the first and third reaction, the energy is being provided by the ATP. All right, so they act as energy transducers. They can act as second messengers. They can act as second messengers. Cyclic AMP, the cyclic GMP. They can act as second messenger, the cyclic AMP and the cyclic GMP. They can act as coenzymes. Nicotinamide adenine dinucleotide, phosphate, flavin mononucleotide, and flavin adenine dinucleotide. All of these are examples of nucleotides being part of the coenzyme. They can participate in one carbon metabolism. They can participate in one carbon metabolism. The classical example being S-adenosyl methionine. S-adenosyl methionine, commonly known by the name of SAM. Commonly known by the name of SAM. S-adenosyl methionine, commonly known by the name SAM. Lastly, they can help as transporters. They help to transport the monosaccharides. For example, UDP glucose or the UDP galactose. They help to transport carbohydrate derivatives. For example, UDP glucuronic acid. UDP glucuronic acid. In addition, sometimes we may find them transporting the sulfates also. The name of the sulfate transporter is very long, so I'll not mention it here, but remember they can work as transporters. They can transport the monosaccharides, the carbohydrate derivatives, and the sulfates. So these are some of the common physiological functions of the nucleotides, of the common physiological functions of the nucleotides. And as you can see, most of these properties are very vital for the survival of the cell. Even those cells which do not have the nucleic acid, for example the RBC, require some of these functions. The energy transducing function of the nucleotide is required by the RBC. The second messenger effect is required by the RBC. The coenzymes are utilized by the RBC. So even if the nucleic acid content is not there, still the nucleotides will be very important for the survival of a cell. Please keep this in mind. The nucleotides are not just the building blocks of nucleic acid. They have large number of different vital physiological function within the cell. Okay, this is the physiological role of the nucleotides, but at the same time the nucleotides can have very critical clinical applications. And for clinical applications we cannot use the nucleotides directly, because most of the nucleotides when ingested will be quickly degraded in the intestine. So what we use are the nucleotide analogs. The nucleotide analogs are slightly modified nucleotides which can then bypass the digestion. Because of their slight modification, they can bypass the digestion. Here I'll not be going into the details of nucleotide analogs, but I'll mention some clinical conditions wherein we can make use of the nucleotide analogs, thereby making them clinically very important. For example, traditionally they have been used in gout. Traditionally, they have been used in gout. Allopurinol is a nucleotide analog. Subsequently, they came to be used in chemotherapy. Classical example being 5-fluorouracil. The classical example being 5-fluorouracil, and using the same property, they have also been used as immunosuppressants. Lastly, recently they have also been used for the purpose of antiretroviral therapy, commonly known as the ART. They are also being used for antiretroviral therapy. We'll discuss the nucleotide analogs in great detail in the pharmacology section because and they form the backbone of the chemotherapy for the treatment of cancers. They form the backbone. So we'll discuss the nucleotide analogs when we are doing the pharmacology section. Okay, so these are the clinical uses of the nucleotide analogs. Now, there is one very important property which is frequently asked in your exam, and that is about the interaction of nucleotides with light. See, most of the substances have a particular absorption maxima. Absorption maxima is the wavelength at which there is a maximum interaction with the light for that given particle. So we have what is known as absorption maxima for every particle. What is the absorption maxima for nucleotides? At what wavelength? At what wavelength the nucleotide will show the maximum absorption or the absorption maxima? What is the absorption maxima for the nucleotides? At what wavelength you will find the maximum absorption given by a nucleotide? What is the answer? What is the answer? What is the absorption maximum for nucleotides? At what wavelength do we see the maximum absorption? The answer to this is 260 nanometer. The nucleotides will show maximum absorption at 260 nanometer. This is the absorption maxima for nucleotides. Now, because the 260 nanometer, due to its low wavelength, has large amount of energy, and this energy will be delivered to the nucleotides when they absorb this wavelength. What happens? The nucleotide can interchange bonds within the nucleotides. When there is interchange of bonds in the nucleotides, there will be interchange of the nitrogen bases, and this is known as mutation. Alright, that is why the light at 260 nanometer is said to be mutagenic. It is said to be mutagenic. Now, when we talk about the light spectrum, we divide it into three groups: the visible light, the infrared spectrum, and the ultraviolet spectrum. In which spectrum does this range fall? Is it the ultraviolet light, is it the visible light, or is it the infrared light? In which spectrum does it fall? In which spectrum does it fall? Anyone? 260 nanometer wavelength of light. In which spectrum does this fall? UV light, infrared light, or the normal visible light? your friend said it is the UV rays. So remember, the UV rays are mutagenic. The UV rays are mutagenic. Alright, the UV rays are mutagenic. Now, one very important question which will be asked is the UV rays are further subdivided into three groups. The UV rays are further subdivided into three groups: less than 280 nanometer, 280 to 315 nanometer, and greater than 315 nanometer. Now, out of these, which is going to have the maximum mutagenic potential? Which is going to have the maximum mutagenic potential? The light which is less than 280 nanometer, the light which is in the range of 280 to 350 nanometer, or the light which is more than 350 nanometer? Question is, which of them is going to have the maximum mutagenic potential? Nucleotides. Nucleic acid is the polymer maximum mutagenic potential. Please note that these are subsequently labeled as the type A, B, and C. So you can give your answer in terms of A, B, and C, which is going to have the maximum mutagenic potential. Okay. (400 to 700 nanometer) is the visible range, and greater than 700 nanometer is the infrared range. Very good. So the correct answer should obviously be 280 nanometer. Now there is a very common question which will be asked in your pathology section. A person is exposed to sunlight which has the mutagenic potential. Which fragment of the UV light in the sunlight has the maximum mutagenic potential? Which fragment in the sunlight has the maximum mutagenic potential? Same three options. What is the answer? Which fragment of sunlight has the maximum mutagenic potential? Same options. What is the answer? Same option. What is the answer? In sunlight. Most mutagenic potential in sunlight. Send me your answers. I'm waiting for your answers. When we say sunlight, the answer changes. And as you rightly said, it is UVB. And the reason is very simple. The reason is very simple. UVC does not reach the surface of the earth. It is completely filtered out by ozone layer. It is completely filtered out by the ozone layer, and that is why in sunlight it is UVB. But if you have an artificial source of the ultraviolet radiation where the filtering by the ozone layer is not occurring, in that case remember the maximum mutagenic potential is for the UVC. It is not for the UVB. UVB is correct for the sunlight, but otherwise the better answer is UVC. Okay, second part is what is known as the hyperchromic effect. The hyperchromic effect. Hyperchromic effect is very simple. What it says, if you have a double stranded DNA and you measure the absorption of UV light by double stranded DNA, you are measuring the absorption. Let's say you get a value of X. Subsequently, you carry out the denaturation. The two strands are now separated, and now you measure the absorption. And the absorption comes out to be Y. As you can see, very obviously the relationship between X and Y. In other words, hyperchromic effect says on denaturation, the absorption of double stranded DNA increases, and this temperature at which the denaturation occurs is known as the melting temperature. The denaturation typically occurs at the melting temperature, and the absorption of double stranded DNA at this melting temperature will increase. So hyperchromic effect tells us on denaturation the absorption of double stranded DNA will increase. This should be obvious. Reason being, when the two strands of DNA are close together, when they are joined by the hydrogen bond, in that case when they are exposed to the light, the UV light, only one of the strands you can see is able to interact with the light. The second strand doesn't show any interaction. The second strand is not showing any interaction with the light. But when you separate the two strands of DNA, both the strands will independently absorb the UV light, and obviously the absorption in this case will be more. So on denaturation, the absorption of double stranded DNA will increase. This hyperchromic effect can be used to determine denaturing effect of various molecules, or it can be used to determine the melting temperature, the temperature at which the two strands of DNA will separate. The hyperchromic effect can be used to identify the denaturing effect of substances, or it can be used to identify what is known as the melting temperature. We call it the melting temperature because the double strand DNA is viscous. When you separate the two strands, obviously the viscosity will be lost. So we call it the melting temperature at which the two strands of DNA separate. Okay. So this is about the nucleotides in general. I'll do a very quick recap. We talked about the nucleotides having the three components: the pentose sugar, the nitrogen base, and the phosphate. We took a look at the nitrogen base. We said it has important properties of being the heterocyclic compound. It has a planar character and it shows the phenomenon of tautomerism. It can be of two types: the purine and the pyrimidine. The purine has a two ring structure. The pyrimidine has a single ring structure. The purines are commonly found in both DNA and RNA, whereas pyrimidines, some are found in DNA, some are found in RNA, some may be found in both of them. The numbering of the purine and pyrimidine rings is different. The purine additionally have four carbons, the nitrogen, the pyrimidine have only two nitrogen. Then about the pentose sugar, we said it can either be ribose or it can be the 2-deoxyribose. The ribose will be present in the RNA, the 2-deoxyribose will be present in the DNA. Because the sugar is also a ring structure, we distinguish the sugar from the nitrogen base using the annotation prime. By using the annotation prime. Alright, so when we attach the sugar to the nitrogen base, we get the nucleoside. When we attach to the phosphate, we get the nucleotide. The various bonds in the nucleotide are the beta and glycosidic bond, the ester bond, and the acid anhydride bond. The two nucleotides are joined together with the help of the phosphodiester bond. The two nucleic acids are joined together by hydrogen bond between the nitrogen bases, which holds the two strands of DNA together. The pairing of the nitrogen bases is as per the Chargaff rule, which says that adenine will pair with thymine and the guanine will pair with the cytosine and double-stranded DNA. This ensures the amount of adenine is equal to the amount of thymine, the amount of guanine is equal to the amount of cytosine. This also follows that in double-stranded DNA, the amount of purine is equal to the amount of pyrimidine. Some corollaries are the different ratios which are calculated from the first few interpretations. Now it can become cumbersome to remember all the relationships. So what we do, we remember the equation 1 where a is equal to t, and we remember the equation 2 where g is equal to c, and wherever required we replace the letters by numbers. After replacement, if the two sides of a given equation are equal, the given equation is correct. If they are not equal, then the given equation is not correct. Talking about the properties of the nucleotides, we said the nucleotides are the building blocks of nucleic acids. They are energy transducers, they are second messengers, they are coenzymes, they participate in one carbon metabolism, and they function as transporters for the monosaccharides, the carbohydrate derivatives, and the sulfates. Clinically, the nucleotide analogues can be used for treatment of large number of disorders. For example, the gout, the chemotherapy. They can be used as immunosuppressants, and they are used for antiretroviral therapy. In addition, the interaction of the nucleotides occurs maximally at the 260 nanometer. This is known as the absorption maximum for the substance. For nucleotides, it is the 260 nanometer. Because the 260 nanometer wavelength has a very large amount of energy, the absorption results in mutation, and therefore the UV light is said to be mutagenic. The UV light is said to be mutagenic. Now the UV light can be divided into three spectrums: the A, B, and C type, and the 260 nanometer lies in the C spectrum. However, in the sunlight, the UVC is completely filtered out by the ozone. So in the sunlight, the UVB has the maximum mutagenic potential. Lastly, we have what is known as hyperchromic effect is seen in double stranded DNA. The hyperchromic effect is applicable for double stranded DNA. This says on denaturation on denaturation, the absorption of double-stranded DNA increases. The absorption of double-stranded DNA increases. Last point, the absorption at 260 nanometer can be used for two things. Number one, quantification, and number two, for quality check, or in other words, the purity of a solution of the nucleic acid. It will be applicable for the RNA, the DNA, oligonucleotides, and even the free nucleotides in the solution. The absorption at 260 nanometer can be used for quantification as well as quality check or purity for a solution of the nucleotides. So this is all about the nucleotides. From here we move on to the polynucleotides. We move on to the polynucleotides. The polynucleotides being the DNA and the RNA. Nucleoside is nitrogenous base plus sugar. The nucleotide is nitrogenous base plus sugar plus phosphate. The number of phosphate may be 1, 2, 3. The nucleic acid is polymer of of nucleotides.